Good morning, students. How are you? I hope you all are safe at home. Today I am going to start Biological Classification Part 1 of 11th Standard. In this session, we will discuss about scientific basis of classification, two kingdom system of classification, five kingdom classification, and also kingdom Monera. So let us start the session. First topic is about scientific basis of classification. There have been many attempts to classify living organisms, but Aristotle was the first to attempt a more scientific basis for classification. The basis of his classification was based on simple morphological characters, and he classified plants into trees, shrubs, and herbs. What are the three? Plant is classified into trees, shrubs, and herbs. He also divided animals into only two groups. That was based on presence or absence of red blood cells in the living organisms. So what was the classification based on? Presence or absence of red blood cells in the living organism. So he classified animals into two major groups and plant into three major groups. After that, Linnaeus gave two kingdom system of classification. He divided whole living organisms into two groups, that is Plantae and Animalia. What are the two? That is Plantae and Animalia. But there was some demerits of this classification. So let us see what were the demerits of this classification. According to his classification, prokaryotes and eukaryotes did not find separate place in the classification. Both were clubbed together. Unicellular and multicellular organisms are also not separated. Besides this, photosynthetic, that is green plant, green algae, and non-photosynthetic, that is fungi, they are also not separated. So further, these two kingdom system of classification did not used for a long time. Later in the year 1969, Robert Harding Whittaker, what was the name? Robert Harding Whittaker, also called as R.H. Whittaker. He proposed a five system of classification. The five kingdom, according to him, were named Monera, Protista, Fungi, Plantae, and Animalia. Now let us see what was the basis of his classification. This is very important. What was the basis of his classification? That is Robert Harding Whittaker's classification. It was based on cell structure, thallus organization, mode of nutrition, reproduction, phylogenetic or evolutionary relationship. So these were the basis for classifying the whole living organisms into five kingdom. What was the five kingdom? Monera, Protista, Fungi, Plantae, and Animalia. Now let us see about the different kingdoms separately. First is about Monera, kingdom Monera. In Monera, all bacteria are grouped, and members of Monera are prokaryote. They may be photosynthetic autotrophic or chemosynthetic autotrophic, but most of the bacteria are heterotrophic. Heterotrophic means They do not prepare their own food, but they have to depend on others for deriving the nutrition. Monera are omnipresent. Omnipresent means they are present everywhere. They have wide range of habitats, means they are present in different dwelling places where they live, survive, breed and flourish successfully. Their wide range of habitats are hot springs, deserts, snow, deep oceans, in the body of plants, in the body of animals, including human beings also. Some are parasite, means they can live inside the host and they harm them. So, through this, we can see that Robert Whittaker's classification was more appropriate. So, that is why we are studying. So, though members of Monera are simple in structure, but their behavior is complex. Now, let us see classification of bacteria. Why? Because Monera includes bacteria. So, we should know that there are different types of bacteria. So, on the basis of shape of bacteria, they are classified into four major groups. They are coccus, bacillus, vibrio and spirillum. Coccus are spherical bacteria, bacillus is rod-shaped bacteria, vibrio is comma-shaped bacteria, whereas spirillum is spiral-shaped bacteria. Here you can observe the bacteria of different shapes, that is cocci. Plural of coccus is cocci. Plural of bacillus is bacilli. And plural of spirillum is spirilla. And plural of vibrio is vibrio. So here you can see that different shapes of bacteria are present. And this was the basis for classifying the bacteria. Here you can also observe that a spirilla having flagella. Further, Monera is also classified into Archaebacteria and so let us see the differences between these two types of bacteria. First, we will read about Archaebacteria. These Archaebacteria are special because they live in most harsh habitats such as extreme salty water areas, and they are called as halophiles, hot spring, they are called as thermoacidophiles, and marshy areas, they are called as methanogens. So you can see here that different harsh conditions these bacteria are present. Archaebacteria differ from other bacteria in having a different cell wall structure. They have different cell wall structure, and this different cell wall structure enables them to thrive or survive in extreme harsh condition. One question always arises from this topic, that why Archaebacteria is special. What is the question? Why Archaebacteria is special? So the answer to this question is that Archaebacteria have different cell wall structure, and this cell wall helps them to cope up extreme harsh condition. So they are special. So due to the special types of cell wall, they are special. One other group of Archaebacteria are methanogens. These methanogens are present in the gut of ruminant animals like cow, buffalo, etc. And these methanogens are responsible for production of methane gas from dung of these ruminant animals. Here you may clearly observe Archaebacteria. Now let us see about Eubacteria. Eubacteria are two bacteria. They are true bacteria. They have rigid cell wall and presence of flagellum if they are motile. Again, this Eubacteria is of different types, that is cyanobacteria, chemosynthetic autotrophic bacteria, heterotrophic bacteria, and one category is Mycoplasma. You can observe a typical Eubacteria. Though Archaebacteria and Eubacteria are structurally similar whenever you observe, but they have different molecular features. So they are different in molecular features. Now we will learn about the different Eubacteria. First is cyanobacteria. Cyanobacteria are also called as blue-green algae. They have pigment, chlorophyll A, and are photosynthetic, so autotrophs. Why? Because they have chlorophyll A pigment similar to the green plants. So that is why they are photosynthetic and autotrophs. These cyanobacteria may be unicellular, colonial, or filamentous also. Means they are having one cell, or they lives in colony, or they are filamentous. They may be marine or terrestrial algae. They may be marine or sometimes terrestrial also. Terrestrial means moist places. Here, colonial forms are generally surrounded by mucilaginous, that is gelatinous sheath, and inside that cyst large number of these cells are present. Some of these cyanobacteria have capability to fix atmospheric nitrogen in special cells, and that special cells is called as heterocyst. What is called as heterocyst? So these bacteria have these special cells, and the special cell is called as heterocyst, and here they fixes atmospheric nitrogen, means they are capable of converting atmospheric nitrogen into soluble form of nitrogen. Example of cyanobacteria are Nostoc and Enavena. Here you can see cyanobacteria. Second one is chemosynthetic autotrophic bacteria. Chemosynthetic autotrophic bacteria are responsible for oxidation of various inorganic substances such as nitrates and ammonia, and uses the released energy for their ATP production, that is adenosine triphosphate. This bacteria plays important role in recycling of nutrients like nitrogen, phosphorus, iron and sulfur. So that is why they are called as chemosynthetic autotrophic bacteria. Third type of Eubacteria is heterotrophic bacteria. Means they have to depend on others for deriving nutrition. This type of bacteria are most abundant and mostly decomposers, helps in decaying the waste. Heterotrophic bacteria are also helpful in making curd from milk, production of different antibiotics, fixing nitrogen in leguminous root nodules, and helps in sewage treatment, particularly treatment of liquid waste, that is secondary treatment of sewage, that is waste water. Some heterotrophic bacteria are causing severe diseases in human beings like chlorella, typhoid, tetanus and citrus canker in plant. Here you can observe the heterotrophic bacteria. Now last one is about Mycoplasma. This Mycoplasma also present in Monera group. So Mycoplasma lack cell wall and they are the smallest of all living cells known and can survive without oxygen. Here it is the figure of Mycoplasma. Now let us see about the reproduction in bacteria. Bacteria reproduces mainly by fission, but during unfavorable situation, they reproduces by formation of spores. They also reproduces by a sort of sexual reproduction in which there is only transfer of DNA from one bacterium to another by a conjugation tube. So with the help of conjugation tube, there is transfer of genetic material, that is DNA, from one bacterium to another bacterium. Here in this figure, you see how there is transfer of genetic material, that is DNA. One will be the donor and other will be the recipient. Okay, so here it is clearly seen in the given figure. So this is all about today's session. Thank you children, and for any kind of doubts please write your comments or questions in comment section.